only need to know 3. So let me explain how we're going to do this. So we have 2 pathways that we're going to talk about, and these are going to be the glucogenic ones. And these will specifically run off of glycolysis and the TCA. And then the other 2 pathways are going to be run on amino acids themselves. So like, I mean, obviously you have essential amino acids, you have non-essential amino acids. So these are basically ones that break down off of amino acids themselves. So I'm going to start off with what you do know. So you guys watched my video last time with the nitrogen pathway, maybe, yes? So I'm going to go over some of the basic concepts of this again because it shows up again. And that's how I hope that you can kind of integrate it into something that you know already. So in the video I said that you can make a bunch of amino acids from things that you have already, things that are already in your glycolysis and some things that are already in your TCA cycle. So the important ones that you have to know are the ones that are pyruvate, and how pyruvates can interconvert with alanine, and how oxaloacetates can interconvert with aspartates, and how alpha-ketoglutarate can interconvert with glutamates. So you know that already. So you know how 3 of them can be degraded. They degraded right into TCA or glycolysis, and they can be created from things that you know already, like these 3 that are in glycolysis and TCA. But before I go into more of the details of this, I'm going to throw a lot of mnemonics at you today because it helps you kind of put things together. So the first mnemonic that I feel is really important to understand and know is going to be PVT Tim Hall. So you probably maybe have learned this when you did the MCATs or anything like that. I'm just going to list them really quickly, and some people, they make a little tiny story about like Private Tim Hall. He's like a war hero, and P stands for phenylalanine. And I'm going to refer back to this chart every so often because it will help you answer questions. It will help you to understand that some of them cannot be synthesized. These are essential. These cannot be synthesized in any shape or matter. These are only degraded. So phenylalanine is the first one. It's the largest one. This is a really big mnemonic. And so on my spreadsheet on Excel, on my my little like YouTube sheets, I put a mnemonics list for you, and hopefully it will be useful to you. I listed all these mnemonics there, and this is what I used to help me answer questions. So phenylalanine, valine, is very valiant. Threonine, if you notice that T is like the 3rd letter of this, and then he has to take a lot of trips around the world. Tryptophan, it makes sure that you don't screw up with like tyrosine or something like that. Isoleucine, so that's like he's isolated from his family and everything like that. Methionine, that's like his method to his madness because he wants to be in history. Let's see, histidine, and then A is for arginine because it's very arduous to be a war hero, and then lysine and leucine. So obviously you know that there's 10 of them that are going to be automatically essential. So technically, we only need to know 10 more in their synthesis because we already got 3, right? So we have 10 that we don't have to memorize. We have 3 that we just figured out. So let me talk about the enzymes that are required for here. This needs PLP. Remember, it's aminotransferase. And remember what your middleman is. What's your middleman? Glutamate. Glutamate is how you interconvert all your amino acids. So there is one amino acid that is really close to your aspartate and glutamate. So that's going to be your asparagine. And then your glutamate, which is going to be glutamine. And you guys probably know how these are synthesized and degraded already. So asparagine, how do you make asparagine? Asparagine synthase. How do you break down asparagine? Asparagine, asparaginase. I can't. Janase. Okay. And then glutamates. How do you make glutamates? Glutamine synthase. Oh, it's a lot of room. And then how do you break it down? Glutaminase. So right at the bat, let's count how many we know. We have 1, we have 2, we have 3, we have 4, we have 5. We know how they are degraded, and we know how they are created. There's going to be, I'm going to decorate this a little bit with other degradation pathways, but for now, I'm just going to let you look at that part first, because these are things that you should know already before you even get into the other pathways of degradation. So that's my first pathway. This is things you should know. And then that's one of them. So the next pathway that I need to talk about is going to be the small amino acid pathways. The small amino acid pathways, these are small ones. They're like serine. Serine is kind of small. It only has like a CH2 plus an OH group. Cysteine, glycine. They're all tiny. Threonine, I'll add that too. And these all stem off of glycolysis. So what happens is, you start off with a glucose. You have a whole bunch of steps. And you're going to get to a point where you're at like 3 carbons. You passed your gap and everything like that. You're at 3 phosphoglycerate. What stems off of 3 phosphoglycerates is going to be a serine. And then the serine, all right, so this is where I need to give a talk about folate. So serine, when it breaks down, it becomes glycine. And we have to talk about this reaction. And we have to talk about this reaction because these are really important reactions. We have to know these reactions. These reactions are going to be dependent on tetrahydrofolates. But I have to tell you about tetrahydrofolates because when I took this course, I was really confused about tetrahydrofolates. So I need to tell you a story about tetrahydrofolates before you go on, because right now, I don't think you know enough to understand, like, there's some reactions that use tetrahydrofolates, and some of them help make the activated forms of tetrahydrofolate. So let's just talk about this. I'm going to go off to the side. We're going to talk about the story about tetrahydrofolates. because this is really important. So tetrahydrofolate, it starts off as folate, and then that's going to be which vitamin? Do you know which vitamin? B9. It's B9, and what happens is it becomes dihydrofolate. And then it becomes tetrahydrofolate. This is irrespective of what I'm talking about right now. This is just how it's formed. Like you get folate from your diet. It gets charged up with NADPH with an enzyme called dihydrofolate reductase. It's the same enzyme for both of them. I'm just going to copy that. Paste that over there. Okay, so let me tell you about dihydrofolate. Tetrahydrofolate is a carrier. It's a carrier molecule. It carries methyl groups. It carries formal groups. It carries carbon. It carries carbon. When it's in the form of THF as it is, it's empty. It doesn't have anything. It's empty. I don't know. I remember as THF, but if he refers, yeah. So yeah, FH4 is the same thing, but I mean, I just use THF, tetrahydrofolate.
I also saw a point. TH4. I don't know if it was typo. TH4.
Yeah. That it's the same thing. Same thing. Okay, so I'll write it down so that. Yeah. So yeah, that's fine. What was it? FH4? He said. FH4? Is that what he said?
Yeah.
Okay. All the same thing. Okay. So we have tetrahydrofolate. This is empty. All right. We charge it up. We charge it up to THF plus some type of group. It's usually like a methylene or 4-mil. It's some sort of thing, not a methyl. Just not a methyl. It could be anything, but not a methyl. This is charged. The reactions that you will see that will use a THF, which makes it from the empty states to the charged states, are the reactions for the amino acids serine, glycine, histidine, and tryptophan. So I like to think of it as like the 4 folate producing ones, the 4 folates. I mean, you could probably make a mnemonic for like SG, H, and T, but like it's important. So you have to know that this makes charged folate. And this is also, not only is it charged, but it's also active. What happens with this folates is that the folates can be stored away. And when it gets stored away, it's in the form of methyl. So watch out. THF methyl is stored folate. And then the THF of anything else is active. This is the important part of it. So where people get confused is this pathway. THF, methylene and formal are used for pyrimidine synthesis. I got it. This is used for DNA synthesis. So, your body needs active folates. But many times when you're not using it, it stores it away. But, sorry, this was not a unidirectional arrow. It's a one-directional arrow. It can only go from active to the stored form. There's no way back. So there's only one reaction that can bring you back to a charged state. This is the one that requires methionine pathway. So you talked about methionine today. I heard from one of your classmates that there was like 5 or 6 slides on methionine. This one uses the methyl group of THF. The other ones make it. That's a really important distinction. So what the methylene pathway does is that it gets rid of the methyl group and then makes a regular THF again. And then this can be recycled again.
So methionine is produced in that pathway?
So the methionine, we'll go over that pathway. But what happens is methionine is produced. Yeah, but I'll have to talk about it a little bit more. But just know that methionine pathway gets rid of the methyl group from folates. Everything else that we're going to talk about, these 4 reactions, are going to put the methyl group on it. So keep that in mind, because I'm going to go back to amino acids. So we saw some of those reactions. Well, we saw that everything except methionine are adding methyl groups onto THF. This one is an inactive THF, and then it makes a THF methylene. What is THF methylene used for? DNA synthesis. Glycine. When glycine breaks down, it will make a THF methylene. What is the THF methylene used for? DNA synthesis. When stored, what does it become? THF methyl. All right. So let's talk about a little bit of the degradation pathway of this. So the glycine, when it breaks down, let me just go over some structure stuff. So serine, how serine has this group. It looks like this. Well, how do you get from a serine to a glycine? Yeah, you need to get rid of OH and a methyl group, right? So that's what the enzyme is called. Hydroxy methyl transferase. Where do you think that the methyl went? THF. Okay. So glycine. Let's look at glycine. So if you got rid of this, then glycine's group is only a hydrogen, right? So yeah, that makes sense. It's glycine now. All right. So now this glycine over here gets broken down completely. This is the only amino acid just poof, it goes away, and it goes away into CO2 and ammonia. But glycine usually has another carbon on it. Where did it go? There it went. Okay. So know the DNA pathway. It's going to be important for vitamin B12 and everything like that. Methylene. Everything in the synthesis is either methylene, a formyl, or something like that. There's another enzyme that changes methylene, formyl, everything like that to a methyl to store it away. What was that? Lysine? Degradation for lysine? Oh, glycine. What do you mean? Oh, no, it's not that. Sorry about that. Okay, so it's called the glycine cleavage enzyme, right? Yeah. So at least those names are easy to remember, right? I mean, because of like a hydroxymethyltransferase, you're getting rid of the hydroxylate methyl to put on PHF, and then like the glycine cleavage enzyme. All right, so let's see. All right. Yes. It makes the tryptophan makes a formal. Yeah, it makes a formal. No, it makes a THF methylene. So yeah, it also makes a THF methylene. I'm pretty sure about that. All right. Are we ready to decorate some of these? Okay, we're gonna decorate these with like just things that kind of stem off of these pathways now. All right, so this is like the basic scaffolds of synthesis, and if you count them, there's 9 of them right now. So let's go to some random things that you just gotta know. I'm gonna make them into a different color so that you know how random they are. Okay, so glutamate can make proline. It can integravert. That's the first one. Tryptophan, since you brought it up, can make alanine. But it actually makes other things too. So I think that when we took it, he went through this whole entire pathway of how tryptophan makes all these things. All you really need to know is just one enzyme. The enzyme is tryptophan pyrrolase. And you need to know that it uses NADPH and it uses O2. And the other products that it makes is going to be niacin. I'm going to make that red because we got tested on that. What vitamin is niacin? Three. Diarrhea, dementia, and death. So that's 3. All right. So there's another one. This is the THF methylene. That's not as important. I just forgot to change the color back to, and it's a little too late for that. And then the last thing is in the degradation it means acetoacetate. All right. So throughout all these, I'm going to tell you a lot of like acetylacetates and then like odd chain degradation, and it's really hard to memorize all those things. So I have mnemonics for those. Okay, so what I'm going to do is I'm going to give you this mnemonic called PIT. PIT are all the amino acids that can make glutatogenic and ketogenic products. So how I remembered it is Brad Pitt is so talented that he can do both glutatogenic and ketogenic products. So bad. All right, so the first one is phenylalanine, isoleucine, tryptophan, tyrosine, and threonine. So by looking at that, we are going to, I'm just going to go over these mnemonics several times so you just get used to knowing them. So, yep. Acetoacetate. So let me describe what glucogenic and ketogenic is. Glucogenic means that it makes anything that's in the TCA cycle or glycolysis. Ketogenic is only acetyl-CoA. So we're going to talk about tryptophan a little bit because we just talked about tryptophan and one of the pathways it makes. So it makes acetylacetate. So right off the bat, acetylacetate, what does acetylacetate get broken down into? Acetyl-CoA and acetyl-CoA. So if it makes acetyl-CoA, is it ketogenic or is it glucogenic? Ketogenic. So why is it glucogenic too? Because I just said PIT is glucogenic and ketogenic. Why is it glucogenic? Yes. Because it goes to alanine. Because if it breaks down to alanine, that's the glucogenic part of it, because alanine can get converted to pyruvate. Does that make sense? So you should be able to take a look at an amino acid, and then you have to be able to say, like, oh, is it glucogenic or is it going to be ketogenic? So, for example, proline. Proline, is it glucogenic or ketogenic? Or is it both? Is it PIT? No, it's not PIT. So, let me give you the ketogenic ones. The ketogenic ones are the two L's, lysine, leucine. So let's take a look at proline. Is proline glucogenic or ketogenic? Glucogenic. Because look at it, there's only one place it can go. It can only go to glutamate. So that's what you want to do to confirm that you know what you're talking about when you actually look at these synthesis and degradation things. You have to see, like, oh, tryptophan, it's both. It's glucogenic and ketogenic, so it has to make something else other than going to alanine. Use that to help you navigate through the exam, because he's going to ask you, it's like, is it glucogenic or is it ketogenic? It's going to be that simple. All right, let's take it a little further. Histidine. There are two enzymes that you need to know for this one. The first one is just going to be histidinase. That's a good enzyme to memorize. It's not too bad. The only thing that you have to memorize about that one is that it uses manganese. And he might have said it in the lecture, but the way that you guys are doing it is a little different than how we did it. Because when we did it, we actually had vitamins on the same exact one. Did he stress the manganese at all? No. Okay. Oh, okay. Well, okay. I'll just leave it there. But histidinase uses manganese. Okay. This makes a phi glue. And this phi glue goes to glutamate. I mean, it even sounds like that's going to go to glutamate. But remember what I was talking about, the folates pathway? What were the four that makes an active folate? Serine, glycine, tryptophan, and histidine. So this one also will use a THF. It's actually, oh damn, I made it too crunched up together. THF. To active THF. I'm trying to remember whether there was any important enzymes there. It's like a phi gluase, right? Right? I guess it's not that important. All right. So I'm done decorating that one. That's it. That's it for that group. All right. I want to continue decorating this one. So we have serine, we have glycine. I have to talk about the other ones. I already listed them for you. You have threonine, and you have cysteine that we have to talk about. So I'm going to draw there's going to be a bunch of steps in between. You guys know that at one point you're going to get to pyruvate. And that's going to be a really important degradation pathway for this. Serine, when it gets degraded into pyruvate. If you guys remember in glyconeogenesis, do you guys remember gluconeogenesis, what the three major amino acids that, the three main molecules that are used for glyconeogenesis work? Do you guys remember that? Lag, remember that? So L is for lactate, A is for alanine, alanine's a major one, and then glycerol, and then the last one, the really minor one, is actually serine. I didn't really put it in. It's a really minor player, but serine is still part of it too, because serine can do this pathway. But I didn't tell you that because I don't think it was in his notes, and it was a really minor pathway. So just something for you to know. The enzyme that does this is going to be serine dehydratase. And the only thing that serine has to do to become a pyruvate is lose an amino group. All right, 2 more to go. Serine can break down to cysteine, and cysteine can also break down to pyruvate. The enzymes that are required to do this are not important, but like the ones I do write down should know because he spent time talking about it. All right, last one. Threonine. I'm going to give you one more mnemonic. The last mnemonic that I'm going to give you today is going to be vomit. Vomit is the mnemonic for odd chain lipid formation. So like basically when some of these amino acids break down, when they break down, they don't break down to perfect acetyl-CoA constituents. They break down into propionyl-CoA or something else that becomes propionyl-CoA. So vomit is going to be valine. O is for odd chains. M is for methionine. I is for isoleucine. And T is for threonine. Let's talk about threonine. Threonine, when it's broken down, breaks down into glycine. And then when it breaks down completely, it becomes vomit. So, what type of carbon chain does it become? It becomes some sort of propionyl-CoA. So try doing this. So I'm going to say serine. Is it glucogenic? Is it ketogenic? Or is it both? Glucogenic. So, all right, so this is how I went through it. So, is it in the pit? No. Is it in the ketogenic? No. It must be only glucogenic. Let's check the pathway. Serine goes into pyruvate. It's glucogenic. You can always confirm your pathways with these mnemonics. That will help you confirm yourself when they ask questions about it. All right, that's 2.
I have a question about it. You said once you go to glycine, you have propionyl-CoA left over?
Yep. And there's actually, let me add this. This is like a threonine dehydratase. Yes?